So welcome to Michael Rosbash's MENDEL lecture. Michael is coming to us from Brandeis University outside Boston, where he's been a faculty member for 40 years or so. Michael is actually from Boston, and then he went to Caltech as an undergraduate. Nuclear RNA and things. Then a postdoc in Edinburgh with John Bishop, who worked on the complexity of DNA genome sequences and RNA transcripts by reannealing and hybridization kinetics, rod curves, cut curves. Then moved on to cloning genes as a faculty member in Brandeis, eukaryotic genes, transcription, and by the 1980s he, together with Mike Hall, who was working on Drosophila circadian rhythms, cloned the period gene, and they began to work on that. I first came close to Michael as a graduate student at Harvard, working on yeast gal4. We had Boston area yeast meetings at Brandeis, and Michael's group was working on splicing in Brandeis at that time. And he went on then in the 90s to look at the mechanisms involved in period and to establish the cycling process involved in the circadian rhythm, the changes in expression level of the circadian proteins, and showed an important transcriptional aspect to that. And that's mostly what he's going to talk about today, except that his title, he changed his subject matter a little. Rather than focusing on circadian rhythms, he's going to talk about RNA editing, which is actually what I work on myself, but he'll be talking about it within the context of circadian neurons in the brain and the fly. So I'm sure if you have questions about circadian aspects, Michael will be happy to answer them. And then, just before we get started, I think I've covered everything. I have a pleasure of presenting Michael with his MENDEL lecture medal. So.
Thank you very much. I see the trouble. No. Let me fix this. Can you put that on there, please? Okay. So, thank you very much for the invitation. It's a pleasure to be here. I don't know how my seminar got. There we are. So I am honored to give one of these MENDEL lectures, and I've never been to Brno before, trying desperately to pronounce it correctly. It's a great pleasure to be here, and I have enjoyed my day as well, talking to faculty as well as students. So I changed my title in part, I must confess, because of Liam and Mary's presence here and their interest in RNA editing. And also I wanted to talk about something new and unpublished that we're doing and we're in the process of writing up, and it's always more stimulating for me to talk about something that we're thinking about and are actively engaged in, rather than giving a review lecture or talking about old published stuff. So if, as Liam just said, if you have questions about rhythms either during the talk or at the end, please feel free to pose them. So this is the title, and I'm going to talk about RNA binding proteins and RNA editing. And the work that I'm going to describe is due largely to two people, two female postdocs, Kate Abruzzi, who's been with me for a long time, really a research associate, and Aoife McMahon, and they have been helped by the other people in my lab whose names are in bold. And so, before talking and putting this in a circadian context, let me say a word about RNA editing, since it's really the background for much of what I want to describe. And so RNA editing, as at least one or two people in this room know very well, is the process by which adenosine is converted by this enzyme, adenosine deaminase, that acts on RNA to inosine, an A to I conversion. And if that inosine is present in the coding portion of an RNA, then it's read by the ribosome as a guanosine, and it will actually change the coding potential of the RNA. And importantly, from the point of view of this talk and my lab's interest, and even from the point of view of circadian rhythms, this process is most prevalent in the nervous system. And so that makes it of particular interest for people interested in behavior, since that means that this process really is likely to have an impact on the nervous system function as well as structure. And shown on the bottom is just an example of the kind of conversion in protein sequence that can happen. And arguably the most well-known and important individual RNA editing event takes place in the GluR2 receptor, in which there's a codon change of glutamine to arginine, and that profoundly changes the accessibility or the permeability of that of the glutamate receptor to divalent cations. And if that doesn't happen, if that decrease in permeability doesn't occur, then the mouse is in big trouble. In fact, the mouse is dead. And interestingly, that is really the most important change that occurs as a consequence of editing in the mouse nervous system, because if you make that change in the DNA, if you change that codon, then the lethality of missing ADAR, missing the enzyme, is actually rescued. So that experiment argues that this is really the most important single change in the mouse nervous system that takes place. And so from a nucleic acid point of view, one of the things that's most interesting about RNA editing is that it requires a double-stranded RNA substrate. And so the change that takes place, the conversion of A to I, occurs within a double-stranded piece of RNA, and much of the recognition of that substrate occurs through these double-stranded RNA binding regions which are present on the enzyme. So shown here are a collection of mammalian ADARs, and in yellow is the catalytic domain, that is the actual deaminase domain which carries out the catalytic conversion of A to I. But these proteins carry additional motifs, of which the most notable are these double-stranded RNA binding domains. And the cartoon, the notion is that those double-stranded RNA binding regions recognize double-stranded RNA binding regions in the substrate, and that's important for determining which adenosine will actually be catalytically converted to inosine, as shown here. And also shown here is the fact that in many substrates where an exon is converted, where an adenosine within an exon, a coding region, is converted, that the paired portion of the substrate is within the intron. So in other words, this is an intramolecular pairing event in which a region of the intron pairs with a region of the exon carrying out this event. And so the kinds of editing events that have been described, in addition to protein coding pre-mRNAs, is shown here, where the glutamate receptor QR site is illustrated. There are repetitive elements which are very important, especially in mammalian and human species, where ALU sequences, for example, are very common substrates for A to I conversion. It's very likely, I might add, that this that this enzyme and this process is part of antiviral defense, so the double-stranded RNAs are recognized, and of course the transposons are dealt with in some ways similar to the way that a viral invader is dealt with, that is to keep transposition at a minimum. And then other double-stranded RNA structures in the genome, such as microRNA precursors, are also subject to A to I conversion, which in some cases will actually convert a microRNA, an A in a microRNA, to an inosine. And so the backdrop, if you like, is summarized here, and that is the recognition of specific substrates, other than the fact that double-stranded character seems to be important, is not well understood. Neither the specificity nor the efficiency is well understood. So why you pick out one A in a double-stranded region and not another A, or why one double-stranded region is ignored and another double-stranded region is converted is not well understood, and why in some cases the conversion efficiency is 10%, and in other cases the conversion efficiency will be 90% in a particular sample. So there are some neighbor preferences. There's a very loose consensus sequence, but not very much known. And so it's with this in mind that we began this project. And I should say, by the way, that my history, as Liam summarized, is really an RNA person at my origin. And so, although I've had a long midlife crisis culminating in being a circadian biologist, I'm trying to return a bit to my youth with respect to this project. So you'll see the idea. So so there are, from studies from my lab and also Rob Reenan's lab, let me just say that there are high throughput sequencing studies in which the brain or the head of the adult fly, in Reenan's case, the bodies of flies, were subject to analysis in which the whole genome was analyzed. And this summarizes the results of those studies. And I think the two most important things to transmit are the fact that there's a much higher level of editing in exons in flies compared to mammals. That may be in part a consequence of the fact that transposable elements are less prevalent in flies than mammals, and as a consequence, the substrate distribution is shifted more toward messenger RNAs in flies than mammals, simply by changing the denominator. And the second conclusion is there's a high level of editing in 3' UTRs. And so, not surprisingly, in flies, as well as has been shown previously in mammals, that editing targets many ion channels and other key neuronal proteins which are altered by RNA editing. It really is a brain and nervous system preferred function, and therefore its targets are the kinds of things that you find in the nervous system. And so here's a set of questions which can frame what I'm going to tell you about for the next 10 or 15 minutes, is what's the function of editing in Drosophila? And of course, one possibility, which is an extrapolation from the GLUR2 example I described to you, is that it tweaks the activity of channel and receptors in cell type specific manner. Everybody know what the word tweak means in English, right? It's bricolage or something. It modulates protein levels by affecting RNA levels. I'll show you an example of that in a moment. Or perhaps it modulates the activity of proteins in response to environmental changes. And I'll say something about that, homeostasis, if you will, in a moment. So here's a piece of data from the Reenan laboratory, which I think is quite informative and perhaps gives a rationale for why the 3' untranslated region of substrates should be such a rich target region for editing. And what Renan's lab did was look at the levels of messenger RNAs in an ADAR mutant in which the single ADAR of Drosophila is missing. And these above the line here are messenger RNA levels which are higher in the mutant, and these are lower in the mutant. And what you can see is that for, and in the colored triangles are substrates which have 1, 2 to 4, or more than 5 ADAR sites in their 3' UTRs. And what I think you can see is that the heavily modified, the substrates with the largest number of sites in their 3' UTRs, their levels increase in the mutant, arguing that the function of ADAR, or a consequence of modification of those RNAs, is to lower the RNA levels, right? So an implication would be that one of the functions of ADAR is to manipulate RNA levels and function negatively to keep them from getting too high. And so here's a second experiment which Liam knows very well, and it's a very sexy example because it's very cool, as you'll see. So the idea is that the amount of editing of a particular codon in a particularly important potassium channel in the octopus is sensitive to the water temperature at where the octopuses were collected. So these octopi were collected at different regions in the world, and as you can imagine, the water temperature here and here is much higher than the water temperature here in the Caribbean, for example. And when you look at the percentage editing here, this editing scales dramatically with water temperature, right? And so therefore the fraction of this codon which is altered is scales linearly with the water temperature. And the idea, of course, is that that channel will be more effective or less effective depending on whether the animal is living in warm temperature or cold temperature and moving around. And so the idea is that editing may be relevant to temperature adaptation of some animals which are cold-blooded, and a nucleic acid concept that I'd like you to keep in mind, and which underlies much of what I'm going to say, of course, is that if double-stranded RNAs are the substrate for this editing event, you can imagine that at cold temperatures, the double-stranded RNA character of molecules is going to be more extreme. It's going to be more double-stranded than at warm temperatures, and consequently you would get an increased editing frequency as a function of that simple change. And of course, this brings me to circadian rhythms, and I'll just tell you that one of the most interesting and least understood aspects of circadian rhythms, in my opinion, least understood, is temperature compensation, as it's called. And a biochemist, this term actually preceded the interest of people educated in biochemistry in the circadian problem. But suffice it to say that this could be called temperature independent, to keep things simple. And what it means is that the circadian period of an animal, a cold-blooded animal, is independent of ambient temperature. So a fruit fly at 18 degrees or 30 degrees, its circadian period is 23.8 hours. So the Q10 of the period is almost exactly 1.0. And so there's no change in the cycle time. And this is almost unprecedented in biology, because anybody who works with an enzyme knows that in general at higher temperatures the kcat of the enzyme is higher until, of course, you get too high and then the enzyme denatures, and then, right? So there's dramatic temperature sensitivity of enzyme activity. And even the cell cycle, another periodic phenomenon, the cell cycle is highly temperature sensitive. The cycle time gets much shorter at higher temperatures than at lower temperatures. So this is a virtually unique phenomenon, and it's not understood how that is regulated. And of course, I'm raising this as a possibility that someone interested in RNA may think that aspects of RNA structure may be relevant to temperature compensation, right, in some way. By the way, there's not a shred of evidence that that's true, but I think it's a good idea. And since there's no evidence, nobody has solved the temperature compensation problem, why not? So, so why else is a circadian biologist interested in RNA editing in addition to temperature compensation? So does it actually affect circadian rhythmicity? If you have a mutant in ADAR, is there a change in circadian properties? Oops, is there circadian cycling of RNA editing? If you look at an editing event, does the percentage of editing change across the cycle? And lastly, how neuron specific is RNA editing? Do different circadian neurons, which do different jobs, manifest different degrees of RNA editing? So I'm not going to show you data for this or this, but the answer to this is yes. Editing does affect circadian rhythmicity. I'm not going to show you data for this. This is a very good experiment, but it's simple. The answer is yes. This is the data are more equivocal, but there are at least a couple of substrates which may undergo circadian cycling of RNA editing. And what I really want to focus on is neuron specificity, because that's where I think we've developed the most interesting story. So here's the beginning, or the introduction, where I would normally start, almost start, if I were giving a talk on circadian biology. And it's this core transcription-centric feedback loop, which we discovered almost 15 years ago. And let me say, I was encouraged to say a word or two about how I became interested in circadian rhythms as somebody who was really a nucleic acid and genetics person. And I will tell you it very briefly. And the answer is, it was strictly through friendship. So I had a colleague on the faculty at Brandeis who worked on flies and behavior and was a neurogeneticist. And I was a biochemist, a nucleic acid person. And we were best of friends. We were both single. We played basketball every day together. So we were 35-year-olds who really behaved like they were 22-year-olds. But of course, that cemented a friendship. And when the time came, when this problem became ripe, and it became time to clone these genes, when recombinant DNA appeared, it was Jeff Hall, I said to Jeff, we can do this, and I think I can help you do this. So we did this together, more or less because we were friends. And of course, one thing led to another, and you start something, and then it becomes very interesting. You know, a cup of coffee suddenly leads to 40 years of marriage and 3 children, right? That's the analogy, right? Everything begins very small, and then you never know where it's going to lead. Okay, so enough philosophy. Back to the circadian biology. So we know that the center of the problem, of the circadian problem, is a feedback loop in which a positive transcription factor composed of 2 basic helix loop helix pass proteins, a heterodimer of clock and cycle in flies, and these are the orthologs, the precise orthologs of these 2 proteins in mammals, clock and BMAL, that these proteins drive the synthesis of their negative regulators, that they drive the transcription of period and timeless in flies and period and cryptochrome in mammals, and those proteins feed back and turn off their own synthesis by directly interacting with the positive transcription factor in the nucleus. And all the bells and whistles, all the details here of how this regulation occurs, that is a delay in the cytoplasm before nuclear entry, a degradation of the proteins, of these proteins at the end of the cycle, all of those bells and whistles cause it to take 24 hours to go around this cycle and for the cycle to begin again. Right? So that's the short version of the center of this problem. And a slide I just added in after conversation with Leon, I want to point out to you that the current view, a paper published from John Hognish's lab just in the last few months, argues that more than half of the genome in mammals is under circadian control. So of the 25,000 genes that mammals encode, more than half of them are under circadian control somewhere. And of course, this varies from tissue to tissue. What's controlled in the liver is almost entirely different from what's controlled in the kidney, with the exception of those core clock genes, which are under circadian regulation everywhere. The timers are everywhere. But the output genes are, of course, tissue-specific, which gives rise to what are the properties in those tissues which will be circadian. And this is a heat map from the liver showing that the mRNA oscillations in the liver, the 4,000 oscillating RNAs in the liver, their peak is all across the cycle. So these are indirect outputs of the clock. So what I want to talk mostly about is the brain. And I point you to the fact that there are about 75 clock neurons in the brain of the fruit fly, which is where all the action happens from a behavior standpoint. So when we stain the brain with an antibody to the period protein, you can count late at night about 75 red nuclei. And if you stain the brain 12 hours later at noon rather than at midnight, there's no detectable immunofluorescence. So the protein levels in the brain are going like this, just as you'd predict, and all of those neurons are beating in sync. They're synchronous. And so the general idea, how did I get that second arrow? So the general idea is that the core molecular clock gives rise to circadian neuron firing, which gives rise to behavior. So the clock is causing the neurons to fire, and then the firing is giving rise to behavior. And the question is RNA editing involved in this process? And this is even more interesting due to current work that we've done just in the last couple of years, where we know, I'm very sorry for this slide issue, where we also know that the firing influences the core molecular clock as well. In other words, the neurons are firing and having output consequences, but the firing of the neurons is also feeding back into the nucleus and adjusting the phase of the clock. And we, over the last decade or so, have discovered that there are separate oscillators which are responsible for the morning peak of activity in the fly and the evening peak of activity in the fly. So this is a typical fly actogram. This is, by the way, very similar for mosquitoes and many different classes of insects. And the animals are active in the morning. They take a siesta in the middle of the day. They're active at dusk in the evening, and then they sleep at night. And notice that the flies anticipate the discontinuous transitions that they're going to experience in the incubator. So the lights come on here and the lights go off here, and yet the animals are active in advance of those discontinuous transitions, right? So it's the clock which is telling them when to be active, not the response to the environmental change. Or to put this another way, anticipation is of a tremendous advantage, probably drove the evolution of the clock. If you look at your watch and it's 7 o'clock and that's dinner time, and you say, it's dinner time, and you're still in the laboratory, that's not such a good thing. But if you can look at your clock at 6:30 and say, I'm due home in 30 minutes, that's anticipating the event and of much greater advantage. So we know that these morning cells consist of these 4 cells here, And these evening cells consist of 5 cells here. And we can manipulate those cells separately and shift separately the morning peak and the evening peak. And if we destroy those cells separately, we destroy the morning peak, leave the evening peak, or destroy the evening peak and leave the morning peak. So from an editing point of view, you'll see in a moment, it becomes quite interesting to ask, this is arguably the smallest, most discrete set of neurons available to ask a question about RNA editing. Is it different in these different neurons? How different is it and why? And so we have, over the last few years, developed very simple methods where we hand isolate neurons which are labeled with fluorescent markers. They're labeled with GFP. The reason we don't use FACS is because we only have 8 neurons per brain. Dissect the brains, 8 neurons. We collect 50 to 100 neurons. 50 to 100 cells will get lost in a fax machine, and so we actually pick out the green cells manually under a fluorescent scope, and then the RNA is amplified and subject to RNA sequencing. And I'll just you have to take it. I'll tell you, and I should say that we've not only looked at morning and evening cells, but we've isolated an outgroup, a non-circadian group of neurons, dopaminergic cells, called D cells here, of which there are about 120 in the fly brain. So the question, we have 3 groups of neurons, 2 circadian groups, 1 non-circadian group, and suffice it to say that the RNA profiles are what you would expect for the genes that we know about. So for example, this gene here called PALE is tyrosine hydroxylase. That's the gene which makes dopamine in the dopaminergic neurons, and you'll notice it has RNA in the D cells, but not in the morning cells and the evening cells. And so of the things we know, these are housekeeping genes. They're everywhere. These are specific neuropeptides which have been assigned to specific cell types. And so the point is we can profile these cells and we get what we think we should. And so now the question is, what happens with editing in these different cells? And so are there quantitative differences? Are there qualitative differences from these different cells? And the reason this is of importance, the entire field has only taken whole brains or large collections of neurons to look at the editing events. And of course, if editing is highly cell type specific, they will be lost in the analysis from the large tissue. Okay, so suffice it to say that there are lots of mRNAs that are edited in these cells, and they're the kinds of RNAs that you would expect to find, just as has been found in previous studies from larger samples. And to just lead you to one of the conclusions right away is that there is a major quantitative distinction between 2 of the cell types, the morning and evening cells, in that they differ at the level of editing that occurs in each of the sites that we find, and that, that the extent of editing in one cell type, and I should just say we do not know why, we do not know the physiological purpose of why this occurs. We know something about the biochemistry, but not the physiology. Suffice it to say, I'll just tell you the result. There's more editing in the evening cells than there is in the morning cells. And so here's a plot or a representation of these kind of data in which the extent of editing for about 250 genes that we can assay with great confidence is shown here on this plot, where these 250 genes are lined up here, and this is the percentage of editing that we observe from the evening cells, a smooth distribution, of course, where some genes have 90% conversion of A to I, and other genes have 5 or 10% conversion of A to I. And those same genes and the percentage of editing is now represented from the morning cells in the yellow dots. And I think what you can see is almost all the yellow dots are below the line, right? Indicating that, as I concluded on the previous slide, that the percentage of editing of the same gene in the morning cells is about half of the amount of editing on average than we see from the evening cells. And our outgroup, which is dopaminergic cells, that its percentage of editing equals the higher values from the evening cells. So when we go outside the circadian biology and ask the question for another group of neurons, what do we see? It's the M cells, or the morning cells, which seem to be the outliers rather than the evening cells. Now, granted, this is not a huge survey of many tissues, but it's a sort of working model. So we have M cells with a low amount of, M cells have relatively low editing levels and are the outliers. And so it turns out that because we have sequence data, we can, of course, look at the ADAR transcript itself in these three sources of cells. And it turns out that there's about half as much ADAR mRNA in the morning cells than there are in the evening cells or the dopaminergic cells. So, you know, I'm a molecular biologist at origin, and we like very simple hypotheses to explain things. And so this suggests that the amount of gene expression for the enzyme itself might explain why there's half as much editing in this cell type as there is in this cell type and this cell type. Yes, sir? So did everyone understand his very strong Irish accent? I have trouble myself, actually. So he asked what time of day was the samples taken from. And so the answer is that our data for these three cell types is pooled 12 samples, duplicates each from 6 times of day. So as you would imagine, we began this study with the idea of looking at editing across circadian time. So we have time points around the clock. And that turned out, for technical reasons, to be very difficult to draw a conclusion. But because we had 12 samples, it turned out to be very good for neuron-specific data. And so the answer is we have a lot of trouble looking at circadian variation, and there either isn't very much and or it's very difficult to ascertain. You can ask me in the questions for more detail. But suffice it to say that we're averaging all the way around the clock. And this conclusion is independent of circadian time, this twofold difference. Okay, so M cells have low editing levels. They have low levels of ADAR. And let me say that there are exceptions to this rule. And so this is biology. And in biology, there's an exception to every rule. And many people study the exceptions, and they become fodder for career and so forth. But there are some genes which show equal or even higher editing levels in M cells. So here's an example here, this gene here, flotillin, and there are 2 editing events in that gene. Both editing events are visible in M cells and E cells, but notice the extent of editing is much more significant in the M cells than the E cells. So the rule is that most genes are less edited in the M cells, but there are a few exceptional genes which seem to do very well and are edited more extensively. And consistent with this idea that these exceptional genes, that M cells are exceptional in some way, those morning cells, is that most of the novel editing events occur in the morning cells. So when we compare all of the editing data in the world that we can get our hands on from all of the various sources, and we look for new events that we have discovered that no one else has seen before, those seem to be predominantly in the M cells. All right, so working conclusions, M cells are the outliers. They have less editing quantitatively, and they have the greatest number of unusual substrates. And so the question is why? And here's the explanation. Here's why. It turns out that when we look at ADAR isoforms, that in M cells, there is a small exon in the 5' end of the gene. So this is an IgB browser. These are the exons of the gene as annotated. And there's an exon here, which is present in some places and not others. And that exon is much more frequent in M cells, these are the morning cells, than in either the evening cells or than in the dopaminergic cells. And to show you this more quantitatively, this is actually a way of, this is an analysis from the high throughput sequencing data in which junctions across exon-exon junctions are quantitated. And in blue here are the M cells, and these are the reads which encompass the 0 to -1 or the -1 to 1 exon. This -1 is the key exon. And notice that these reads are much more frequent in M cells than in E cells, right? So there's this particular splicing event which is much more frequent in the ADAR transcript, and that addition of that exon -1 adds 21 amino acids to the N-terminus of ADAR. So this is the most N-terminal double-stranded RNA binding domain, and added on to the end of the protein, this is the normal start, added on in this -1 start is 21 amino acids, right? So the actual enzyme in the M cells at a much higher frequency than in E cells or dopaminergic cells gets an extra 21 amino acids added to its end due to alternate splicing, right? And it turns out, and this summarizes a lot of data, but suffice it to say that if you make those two protein forms and put them in tissue culture cells, now either the long form or the short form, then much to my amazement, the long form has about half as much ADAR messenger RNA as the short form in tissue culture cells. So these are two independent stable lines of the short form, the form that's prevalent in the E cells and the D cells, and this is the long form that's prevalent in the M cells, and when you put those in S2 cells, you get about half as much mRNA from this form. And this is the kind of plot I showed you before, that is that there's less editing for most of the substrates in the S2 cells in response to the M cells, in response to the long form, the M form, than there is in response to the short form, the E form, or the D form. And I'm not going to belabor this, but suffice it to say, there are also a few substrates which are exceptions to the rule, namely, which are specifically edited in the tissue culture cells in response to the expression of this long form here, right? So the conclusion is that RNA editing affects circadian period. I didn't show you this. M cells show less editing. There are some exceptional sites with higher levels of editing. And the quantitative and qualitative difference between neurons is due in part, at least in part, to mRNA splicing regulation, which gives rise to differences in the ADAR levels and also a different RNA binding region, we speculate, that is something different next to the double-stranded RNA binding domain, which then can recognize different substrates because of the change in the protein. And of course, a implication here is that ADAR levels actually determine editing frequency. Or to put it another way, ADAR is rate limiting for the amount of editing among the different kinds of things. Because if you just manipulate the ADAR levels, you output, in general, with exceptions, the amount of editing. Okay, so, and these are the two most important things I just emphasized. Okay, so in my remaining time, and I will try and keep on time, I want to describe a second project whose only connection is that it exploits our interest in ADAR. And it is a new method that we have devised, and it's called TRIBE, which stands for Targets of RNA Binding Proteins Identified by Editing. And the motivation for devising this method is very simple in the following. If you're interested in neurons, and in particular small numbers of neurons and what they do, it's extremely difficult to do biochemistry with such material. Right? It's very simple. If you only have 4 cells per brain you're interested in, and if you have to dissect the brains to access those 4 cells, you can't do any biochemistry. You can't do an immunoprecipitation experiment. You can't do any of those kinds of things. So you need to figure out a way to circumvent that problem if you're interested in characterizing what goes on there. And this is the idea. So for instance, if we would like to know what the targets are of a particular RNA binding protein in a particular neuron, then the strategy we took was to ask the question, if we took the catalytic domain of ADAR and threw away the double-stranded RNA binding domains and just took the catalytic domain and made a fusion protein, could we recognize its substrates based on this particular strategy? And for those of you who are either sophisticated or have stumbled across this in your studies, this is very similar to DAM-ID, a strategy to look at the position of DNA binding proteins by attaching a DNA methyltransferase domain to those DNA binding proteins, except this is for RNA. So the idea is initially, at least, to do this in cells, do this in tissue culture, induce expression of the fusion protein, and then look for new editing events. And so, here's the initial experiments done in tissue culture cells now. And the reason we do these initially in tissue culture cells is then we can do biochemistry and compare the results and ask, what do we get, right, before doing anything in neurons. And so it turns out that there is no editing, no endogenous editing in S2 tissue culture cells because those cells don't express ADAR. So when you sequence mRNA, there are no inosines, right? So that's good. That makes the experiment easy. And suffice it to say that if one expresses this fusion protein in a stable line in which this RNA binding protein, HRP48, is fused to this catalytic domain, and this is uninduced with copper, and this is induced with copper, then you get a big increase in the extent of editing, arguing that, of course, at least the catalytic domain is functional there. And, of course, it has specificity, because if you do the experiment twice, then you get a good reproducibility in both the genes which are edited, the mRNAs which are edited, and the extent of editing in the various mRNAs. So that's good. This works. And then the question is, are these real targets of this RNA binding protein? And the only way to do that is to compare with a biochemical method. And the state of the art for doing this, not without its problems, but the state of the art nonetheless, is this technique invented originally by Bob Darnell and his colleagues. And then there are many variations on this general idea. But you UV cross-link cells in vivo, so the UV light is shined on the cells, and then you purify the protein and analyze the little piece of RNA, which is covalently attached to the protein after purification. And so here's an example of data for the best known target of this HRP protein, which is the heat shock protein 83. This is from the literature. This is a decay element in the 3' UTR of HSP83, which is known to be bound by HRP48. So these are the reads of mRNA from mRNA expression, and these are the clip data. These are the reads from the biochemical experiment, the clip experiment. They're exactly where they should be. for HRP48, and these are the editing events. I'm sorry that it's very hard to devise a metric that shows you both where they are and the frequency of editing, but suffice it to say that the only place in the whole gene which shows editing events is right underneath, is in the same location as the clip data. And so if you now look across the genome wide at HRP48's preference for editing, and you compare clip genome-wide with tribe genome-wide, then the conclusion you reach is that the 3' UTRs are really the favorite location in general for editing events and or HRP48 binding by clip analysis. So here is the distribution on a scale from 0 to 1 of the length of messenger RNAs which are coding 5' UTR or 3' UTR in a fly. Right? So the 3' UTRs of flies are about 15% of the length of the RNA, whereas maybe 75% of the length of the RNA is coding region. And here's what tribe and clip give you for the percentage of the editing events which go into the 3' UTR. Okay, so far so good. And here's the overlap between target genes as defined by clip and tribe. So this is genes, the distribution of genes by clip enrichment, and this is these are the genes which are identified by tribe. And the point here is that the top clip targets, the 100 genes which are the top clip targets, are grossly overrepresented by tribe as compared to the bottom clip targets. So, you know, this is the numbers: 2.8% down here, 20% in the middle, and 50% over here in the top clip targets. So the idea is there is decent but not perfect overlap between these two methods. And of course, clip itself is a method with both false positives and false negatives. So the point here is not that either one is better than the other necessarily, but that it provides a complementary approach for this kind of problem. So here's protein number 2 of RNA binding protein number 2 of interest to neuroscientists worldwide, because this is the project of the Fragile X gene, FMRP, and it's involved in translational repression and also in regulation by neuronal firing. And it regulates a number of different processes: mRNA stability, mRNA transport, translation, synaptic translation under synaptic control in dendrites, etc. And so we made a fusion protein of FMRP with the catalytic domain of ADAR. And just like the experiment I showed you before, we can get a lot of editing events from that fusion protein when we induce the protein in tissue culture cells. I think I have, there we are. Okay, and so this is a particularly useful comparison with what I showed you previously, and this is a particular gene which is a efficient substrate of FMRP and of editing, and these are the reads within the gene, and this is the coding sequence. And what I hope you can see here is that the editing events here are predominantly, if not exclusively, in the coding region now, not in the 3' UTR. So these are clip data from the Darnell lab in the mouse ortholog of this particular fly protein, and notice all the clip reads predominantly in the coding region, and sure enough, our editing events are exclusively in the coding region as well. So a completely different distribution of reads. And sure enough, when you look in a metagene analysis, now the tribe data is predominantly in the coding region, just as the mouse clip data is predominantly in the coding region of the gene. And so here's the clip data, and here's the data for HRP48, which is predominantly in the 3' UTR, and here's the data for FMRP, which is predominantly in the ORF, in the open reading frame. So you can see that the protein that's attached to the catalytic domain of ADAR is determining where this happens. And of course, the thing that's quite interesting about this is that FMRP is supposed to be recruited to particular messenger RNAs, not only by its target specificity in the mRNA, which is unknown, I might add, but also by virtue of an association with the ribosome, hence the restriction to the open reading frame. And so because these experiments have only been done in flies so far, we are limited to tribe targets in the fly which have clear mouse orthologs, which have been analyzed in the brain, for example, by the Darnell lab. And suffice it to say that when that's done, that the tribe targets are highly enriched for clip rankings, which are the most efficient or highest clip targets. So tribe targets are enriched for higher clip rankings. So the same conclusion for these two different proteins, that we can validate this with clip, and that they agree that HRP48 preferentially binds the 3' UTR, FMRP preferentially binds the coding sequence, and there's good but not complete agreement on exactly which genes are the targets. So to wrap up, let me just say, and I'm going to do this very quickly, that we can now go back to the specific cells, which we've only done so far with HRP48, and we can ask the question, can we identify targets in the small numbers of defined neurons using this editing technique? And of course, this is exactly what I showed you before in the first part of the seminar. We pick out the specific neurons with the fluorescent labeled approach, and then we have used 3 different sources of cells that has expressed the fusion protein either in the morning cells and dopaminergic cells or pan-neuronally. And in each of these cases, within the neurons, we get a big increase in editing events when we express the fusion protein in those cells. And I should say, by the way, now, of course, because these are neurons, we don't have zero background, right? Because the tissue culture cells express no ADAR, but of course these neurons express ADAR, as I showed you previously, and so we have some background to contend with, but nonetheless, overexpression of the fusion protein greatly increases the extent of editing that we see. And the results we get, and I'm going to go through this quite rapidly in the interests of finishing within 60 minutes, is exactly what you'd expect. That for some genes, many commonly expressed genes are edited in all 3 cell types. So these are the reads of the genes we get, and suffice it to say that we see editing events here in all 3 cases for some of these genes, right? So in other words, we can detect editing events in excess of what the cell normally has in the absence of expressing the fusion protein in all 3 of these cases. And so also for cell-specifically genes, that is genes which are only expressed in certain cells, so this neuropeptide is only expressed in the morning cells, and we see an editing event only in the morning cells. Of course, it's not there in the other cells, and in the case of this gene here, it's only expressed in dopaminergic cells and TH cells, and we see editing events there. And of course, the most interesting result, the one that we were trying to assess initially in developing this, is that for some commonly expressed genes, they are indeed differentially edited, right? So these genes here are present and expressed in all 3 cell types, but they bind, the interpretation is they bind the RNP protein selectively only in 2 of the cell types here or in 1 of the cell type here. So in other words, the RNP protein does show selectivity in the different cell types based on the fact that the editing events only occur in 1 place or in another place. And that kind of result, fortunately or unfortunately, cannot be checked yet with a orthogonal technique because we can't do biochemistry and we don't have another way of assessing that. But I think it's not surprising, and it's what we designed the technique for. So in conclusion, TRIBE can identify cell-specific targets of RNA binding proteins. There's overlapping specificity in commonly expressed genes. And then even in uniquely expressed genes, there are editing events which occur specifically, of course, for these genes, which of course then can't be accessed or evaluated in any other way. So a summary of this now is that we can identify RNA binding proteins and their targets by using editing, by using this fusion strategy. TRIBE is capable of identifying cell-specific targets. And to just finish on a nucleic acid-centric point, and I have one more slide to expand on this point. It may be that transient double-stranded RNA structures may be marked by the ADAR TRIBE fusion protein. And so what I didn't tell you is that there is good data in the literature that even without the double-stranded RNA binding domain, that the ADAR catalytic domain actually needs a double-stranded substrate in order to convert adenosine to inosine, that it actually must flip the A out of the helix in order to do this job. And so the question is, if that's really the case, how can we have a general technique? Is all RNA structured? Why does this work so well and in such a reproducible way? And so I think my own view on this is summarized in this slide here, that I think one has to think about in vivo RNA structure, even the structure within translating ribosomes, that is along an open reading frame, as really quite dynamic. So this is the reminder that I just mentioned, that ADAR catalysis requires a double-stranded template. And as a second reminder, there's a very important paper published in 2014 by the Weissman Lab, which argued that RNA in vivo, messenger RNA in vivo, was really unstructured or largely unstructured, because using single-stranded probes of structured dimethyl sulfate, that they could access essentially all mRNA by this means. And so I think the fact that we can capture or tag almost many different mRNAs with the ADAR catalytic domain argues that these structures are breathing and forming all the time. And of course, we can capture them anytime they form, and the protein is permanently present there. And so even structures that form infrequently. And so I think an implication is the structure is highly dynamic. And a further implication of this, which speaks to the double-stranded RNA binding domains which are present on the normal ADAR protein, is that perhaps these domains capture transient structures and don't actually search for structures which are permanently or irreversibly double-stranded, right? So it suggests a much more dynamic view of RNA in vivo, which is breathing all the time. And of course, this also explains why the Weissman experiment works, because of course, if the RNA is extended at some point, then it can be a good substrate for the single-stranded probes. And similarly, if it's double-stranded at some point, it's a good substrate for ADAR. And with that, I'll thank you for your attention and be glad to take questions. Thank you very much. Even on circadian biology, or perhaps I should say especially.
I'll pick up the questions myself. If there was part of a thermometer or some system that controls the temperature compensation of the circadian rhythm, then an intermediate, as well as maybe having a longer or shorter day, might be temperature sensitive in its rhythm.
Right. So I was actually, I was not suggesting that ADAR itself was part of the temperature sensing mechanism, but that RNA structure might be important for responding or sensing temperature. So that was the sort of simple idea. So, for instance, if you had two different competing structures or two different RNA molecules, those molecules could be part of a system which would simply be responsive to temperature in a way which might be read out. So I wasn't thinking about it in terms of ADAR necessarily.
Quite a few editing sites show a very high level of temperature dependence.
Right.
It's a small subset of editing sites that show a high amount of temperature dependence.
Right. So you. Right, so you're right. So we discussed a little bit the background for this at dinner. I think it's possible that in mammals, the requirement for really a very strict, stable structure might be more the case, and consequently would render the system less sensitive to temperature. And this experiment really hasn't been done in flies terribly much yet.
I better ask other people for questions.
Yes?
Dalibor.
The crystal structure of ADAR known, for example, with some of the substrates?
So you were just telling me about, there's one crystal structure. You could take this question better than I. Is there a crystal structure of ADAR with its substrate?
There is a structure coming with a substrate, but it's a more artificial piece of substrate from yeast. It's not a natural substrate.
But it's a double-stranded RNA. It's a double-stranded structure.
That's Peter Beal and UC Davis.
Right. And I think the biochemistry done many years ago now by Brandeis argued, the original paper argued that and that only used the catalytic domain, argued that it really needed a helix as a substrate. And I can't remember what the data are for the fact that A is flipped out when the conversion takes place.
Well, Bill sees it now in the co-crystal in that substrate. Okay, and the other data was various probing, mostly done again by Beal, showing increased nuclease sensitivity, I think, or something around the edited base, and fluorescence or spectroscopic evidence that it was flipped.
Right. That notion's been around a long time, but I can't remember. I mean, before a recent crystal structure, but I can't remember quite where it comes from.
Yeah.
So I was wondering about so if you have some kind of affinity for the RNA already with the deaminase domain, then you will lose the specificity that is given by the protein that you are trying to identify, for example. Mm-hmm. And so, yeah, I mean, how do you deal with this problem, and also what would be, I'd say, a control for that?
Right. So you ask a good question, something we worried about, and I think the prediction is very simple. If you have two different fusion proteins, which we do, right, which we've studied, and the number of targets in common would suggest that ADAR is directing those targets, right? So you have two fusion proteins, they have two different RNA binding proteins, but in common is the ADAR catalytic domain. So prediction, if 80% of the targets are in common, then the ADAR catalytic domain is a candidate for determining those targets. If 10% of the targets are in common, maybe those are due to the ADAR catalytic domain, but that's a minority of the number of targets. And the number we see in common is very small. So that suggests to us that ADAR is not determining the catalytic, the substrate specificity. I think perhaps inherent in your question, a more subtle view is when you have multiple RNA binding domains on a protein, who determines specificity and how does general affinity contribute to specificity, right? So for instance, one presumes that the affinity of ADAR for nucleic acid would improve the ability of the RNP protein to bind, and might it also change its specificity, right? That's and all we, you know, you need a complementary method to address that question, and all we can say is that from the CLIP experiments that the overlap is pretty good. Maybe the fact that it's not perfect speaks to some extent to that possibility. That's all one can say about it.
No, maybe on a more, I have to say, more molecular and structural level, maybe a control could be, for example, to have a control with a protein with, for example, many RRMs before, so RNA binding motifs before that are known to be very specific for one RNA, for example, and see if you can really pull down only this one.
Right. So the only thing I could say in experiments I didn't show is that HRP48 has been shown to have a consensus sequence, and that's been shown both by meme analysis of binding sites and also a CellX. There's a CellX experiment that's been done with HRP48, and we get that site in our editing sequences. Right? So it's at least good enough to show the specificity that's been previously defined by HRP48. I must say it's not quite as good as previously defined, but it's not too bad.
Other questions?
Please, could the RNA editing lead to alternative splicing of the RNA?
So the answer is the literature has several examples of an interplay between splicing and editing. And our view is that editing occurs co-transcriptionally in the nucleus predominantly, just like splicing. And so those two processes naturally talk to each other, and they can talk to each other in two broad categories of ways. One is the complexes can actually interact, interfere, complement each other, or the actual editing event itself could then influence a splicing site, and there's examples of that as well, where you either create or destroy a splicing site based on the actual editing event.
I'm trying to think of an example of RNA being used as a thermometer in a process. Are there systems in bacteria that are thermometer-like with RNA structure?
So we've as I hinted, this particular topic takes me back to my assistant professor days, and one of the experiments we did from those times was to create a temperature-sensitive splicing system in yeast, where we hid the 5' splice site in a double-stranded stem, and of course, if the stem was long, then there was no splicing because it couldn't be recognized. But if you made the stem shorter and shorter, you could make the stem to be sensitive just in the nice range for yeast between 20 and 35 degrees. And so you could make a temperature-sensitive splicing event just by creating a duplex, which was 8, 9 nucleotides. I forget how long it was. It's so long ago. 8 or 9 nucleotides. And so that's another reason why this kind of structural work in yeast made me a little skeptical about the view in the Weissman paper that RNA is unstructured in vivo. It just seemed to me not compatible with the literature, the history of the literature. Yeah.
Yeah, I was wondering maybe more on the molecular mechanism for the variant of the ADER spliced in the M cells.
For what?
The spliced variant in the M cells. So these 21 amino acids, do they contain a new property or domain, or they affect the activity of the ADER?
We think that the protein, the simplest interpretation is they either affect translation or they affect the stability of the protein. Because the RNA levels, the mRNA levels, fit the amount of editing we get. So the correlation we have is how much mRNA you have and how much editing. And since that fits, I think you, and miraculously, when we transfer the long and short forms to S2 cells, we get a factor of 2 in the amount of mRNA and the amount of editing. So the simple idea is either that ATG has an effect on translation efficiency, for example, or that's related to protein, somehow related to RNA turnover, and so the mRNA levels end up being a factor of 2 different, and so that deals with the efficiency business, right? So, but the other subtlety is specificity. And now you have an extra 20 amino acids right next to an RNA binding domain and a whole bunch of new substrates which are recognized, and presumably that's really has to do with RNA protein, the RNA protein interactions. That's the implication there. And for there, it would be great to have structure of a co-complex with RNA of the 2 different forms to see what it does to RNA binding. Pretty good for a circadian biologist, wouldn't you say? Anyway.
More questions about circadian rhythm or anything else?
There's somebody here.
Yeah.
Today, a lot of mathematical models of circadian rhythms exist.
They do.
What is your opinion on computational modeling in this field, in general, in biology?
Oh my goodness. Do I have to answer that without a drink? Can I? So, I am a muted fan of computational biology as it applies to circadian rhythms, and to give you, and I'll explain why. I think there are some very, very simple fundamental features of timekeeping that are not well agreed upon, and they would direct modeling in a completely different way. So, for example, right, the modeling of circadian biology is based on limit cycle oscillators, and the whole math comes from. And before we knew anything about proteins and mechanism and nucleic acids, all of the data fit a limit cycle oscillator. And to my mind, the key feature of a limit cycle oscillator is that you have one rate limiting step, which is determining the speed around the cycle, right? That otherwise, the whole thing doesn't make too much sense. And I think in the last 10 or 15 years, one has to think about the circadian cycle more like the cell cycle, where you have a transcription phase, and then you have a entrance to the nucleus, a repression phase, you have a degradation phase, you even have a translation phase, which follows the transcription phase. And each of these takes a certain amount of time, and they add up to about 24 hours, And I think that a key question is, do they have a single rate limiting step? Is there one process which is rate limiting all the way around the cycle, or is there a collection of rate limiting steps around the cycle? And of course, this has implications for everything, including temperature compensation. Because of course, if you have different steps that are different rate limiting processes that are governing the speed with which you traverse a certain portion of the cycle, then you need different temperature compensation mechanisms to govern the different steps, and it's not the same. You have to act on the rate limiting step, right, in order for temperature. So the fact that we don't know, and nobody agrees on, is this what I just said is not well accepted. It's not radical, but it's not well agreed upon either. So I don't know how you model. I mean, you could model one way or the other and so forth and so on, but I don't think it has contributed too much to the way we think about the problem and made predictions which then, can be tested and so forth and so on, So I think it neither, right, here we are with Mendel, right? So what's a theory should have one of two properties. It should either have great explanatory power, right? Evolution. Not many predictions, great explanatory power. Or it should make predictions which can be tested, right? One of the two. So I haven't seen computational biology do either of those two things yet. Now I really need a drink.
Any more questions? We'll wrap.
Someone want to take issue? Someone could take issue with me on that last statement. I have no problem. I'm not...
There's refreshment.
There's refreshments out there? Okay.
Well, I think Mendel would have loved this talk, actually, if he'd been here. He went into beekeeping in the end, apparently. But thank you very much, Michael. That was very nice.
Pleasure. That's right.
It's hard to get something that is temperature sensitive.
Well, so if you had asked me that question